So hi everyone, thanks for the introduction. My name is Peter Contos. I am now an ex-PhD student at Trobe University in Bandura. So I finished my PhD research last year, which is looking at how we can rewild invertebrates and microbes to improve diversity and function, specifically on degraded farms. So just before I start, I'd like to acknowledge the traditional owners of the land on which my research was conducted, the peoples of the Kulin Nation and Turungaran clans, and just extend my acknowledgement to any First Nations people here with us today. So yeah, as I mentioned, our project was in the Strathbogie Ranges, and we'd have study sites that sort of look like this, where we've got a degraded pasture, and a farmer has gone out to try and restore that degraded pasture for diversity and function. And they'd usually go out with groups like local land care, plant a bunch of native species in the hopes of regrowing a mature forest that animals from surrounding remnant areas can then recolonise and use as habitat. So restoration is just generally a way to increase diversity and function in some of these sort of more degraded areas. But an issue that we were noting with our project, and I guess more generally just restoration projects broadly, is that they often exist as a bit of like an isolated island of habitat surrounded by a non-habitat area. So in our case, we had these like really good pockets of restoration on these farms, which were good for species, but they'd be like immediately surrounded by non-native pasture that a lot of animals didn't really like to live in. So there wasn't much connectivity between our remnant sites and our restoration sites. Very simple diagram, but if we look at it like this, we had our sort of species-rich remnant site over here, and it was really geographically separated from our restoration site by this big barren farm pasture. No surprise, but invertebrates, they're small, they often don't have wings. They're really bad at actually dispersing from one site to the other. And a little bit more surprisingly, microbes too kind of differ in their ability to disperse from site to site. Some might be good at moving to new areas, whereas some might be quite poor. Which means that these species we were finding were often struggling to get from these really, biodiversity rich remnant sites and actually recolonising these restoration sites. And what this often results in is in restoration areas, you can unfortunately find less species. And because invertebrates and microbes are so important for function as well, when you have less species relative to remnant sites, you can often find reduced ecosystem functions as well. So what we tried to do in our project was we tried to fix this issue by actively rewilding invertebrates and microbes. And we did this using a pretty simple methodology. What we did was we just went to these really like biologically rich remnant sites. We took a bunch of soil and leaf litter from these sites. And in that soil and leaf litter, there would be the entire community. We packed it into the back of a truck, just drove it over to the restoration area, and then just transplanted it there. So that was our rewilding event. Yeah, it's kind of simplistic, but yeah, that's what it is. It's pretty straightforward. A lot cheaper than some of the big fence and removing cat sort of projects, but I guess that's the benefit of working with tiny little things that people don't care too much about how you move them around. But yeah, this is a really unique form of rewilding, I'd like to think, because a typical project might focus on trying to reintroduce just one or a few locally absent species. But within that leaf litter in the soil, there's potentially thousands of different species that we're essentially reintroducing into a new area. So yeah, as I mentioned, our study sites were in the Strathbogie Ranges in central Victoria. We worked really closely with the Goulburn Broken Catchment Management Authority to identify farms where landholders had revegetated sections of their farm at least 20 years ago. So we were targeting really mature restoration sites. And we had 3 treatments. So we had a remnant site, which was a conservation area, and also the source of the leaf litter in the soil. So this is sort of like our target ideal community. We had a rewilding transplant site, which was obviously a restoration site with a transplant. And then we had a control restoration site, no transplant. To sample invertebrates in the soil and the leaf litter, you use these funky looking things up there that kind of look like UFOs. They're called Talgren funnels. And what you do is you take leaf litter and soil, You put them in these funnels and it sort of flushes the invertebrates out into a collection tray. And to look at microbes, what we did was we took some soil samples and did some DNA metabarcoding to identify the specific bacterial and fungal species that were present in the soil. And this project's been running for quite a while. So the first time the communities were sampled was in November 2018. And that was taken as a baseline measurement, so that's before we actually did any of the transplants. Just to gauge, I guess, what the community looked like before the actual rewilding event. And then we took some samples 7, 18 and 27 months just after that first transplant event. So if you've never had the joy of working with invertebrates and microbes, they're a very complex group to study. They're super diverse, especially the ones in the soil and the leaf litter. Lots of species in there are undescribed to science, and we're not really sure of their functional role and what they do in the system. So to make it a bit easier, we generally break it into 3 groups. So we've got our macroinvertebrates, which are greater than 5 millimeters in size. These are the things you're probably a bit more familiar with. So it's things like cockroaches and millipedes and beetles, that sort of stuff. We have the mesoinvertebrates, which are less than 5 millimeters in size. And then obviously we've got our fungi and our bacteria too, and that sort of makes up the soil and the litter community. So in our project, we looked at all of these groups, but I spent quite a lot of my time focusing on the mesoinvertebrates, which contain a lot of tiny little things like beetles and wasps, which will live their entire lifespan within the soil and the leaf litter, and also things like mites and springtails. But if we're talking about just like, I guess, where the diversity in the invertebrate community sits, it's this group. So this group is hyper diverse, super abundant, very overlooked. I mean, even when I talk about them to other entomologists, they sort of roll their eyes when I talk about mites and springtails, because they're just, I don't know, to the naked eye they're a bit indescript. They sort of just look like little brown specks running around on the surface of the soil. But when you throw them under a microscope, they're really functionally diverse. So that prostigmatid mite up the top there, that's sort of like the top order predator in the system, sort of like the lion. And then those funny looking globular things down the bottom, they're really important prey species and detritivores in the system. So there's your orobatid mites and your springtails. So yeah, very diverse. It's sort of unrealistic to expect that when you move so many species into a new area that every one of them is going to establish. So we were finding throughout our project that not a lot of these groups actually did establish in their new area for a lot of different reasons. But one group that definitely did were the beetles. So this graph just shows the number of beetle species across treatments and across time. And you can see in that first grouping, so the pre-rewilding in the baseline measurement, the number of beetle species in the control and the rewilding site are the same, and importantly, they're lower in comparison to the remnant sites. But as soon as we initiated that first transplant event, so from 7 months onwards, we were getting significantly more species of beetle in the rewilding sites relative to the control sites. And importantly, the numbers of beetles were at the same level as the remnant sites. So what this is suggesting was that we were successfully reintroducing a bunch of remnant beetle species into those rewilding areas. And we were finding that was a lot of these groups, a lot of these beetles were coming from this group called the Trobe beetles, which are these sort of tiny little beetles that are really important predators in the system. So they're actually very functionally important, and they'll suppress a lot of the prey populations. And we were thinking that these were missing from the system, and our transplants reintroduced them. So yeah, species diversity changed, which is, I think, a cool result. But what about function? So rewilding projects, as we sort of have heard from today and probably will for the rest of the day, they often focus on changes in function after you've reintroduced a species. So if you think about, I guess, typical examples from overseas, the reintroduction of wolves into Yellowstone National Park, that was initially done for conservation purposes, but that process of reintroduction also reintroduced the process of predation into the area, which had really strong effects on herbivores, which then had really strong effects on plants. So there's a lot of flow-on effects when you reintroduce these quite functionally important species. Closer to home, as we've sort of heard from Derek earlier this morning, we tend to focus on things like bilbies and bedongs in our rewilding projects. And these dig up the soil for food, and this has really strong flow-on effects to soil processes, and that changes soil communities too. So in our project, we looked at a function heavily driven by invertebrates and microbes, which is the process of leaf litter decomposition, which is this really important process where leaf litter is decomposed and consumed by invertebrates and microbes and then recycled back into the soil as nutrients for plants. So it's a really important process for above ground plant communities. It's a really complex process as well. So depending on the season, it might be driven by invertebrates or fungi or bacteria. Sometimes they'll work with each other, sometimes they'll work against each other. And at the same time, you've got things like temperature and humidity also playing a role as well. So to measure this, we constructed these bags that contain dried leaf litter, and we weighed them, and then we put them out in the field for 3 months, and then we brought them back in, and we weighed how much of that leaf litter was actually broken down and consumed by the invertebrates and the microbes. So that loss in weight is indicative of how fast the leaf litter is actually decomposing. So we measured this 3 times and found a little bit of a seasonal effect going on. So depending on the season, you might have a strong effect of treatment or it might drop off. But we found that when you looked at it overall, there was significantly faster rates of decomposition in our rewilding sites relative to the control sites. And then when we threw that all into a big model, what it was saying was that decomposition was driven by abundances of invertebrate detritivores. So that's things like trilobite cockroaches, millipedes, other things like that too. So in our rewilding sites, we were finding greater numbers of these detritivores, and they were enhancing and speeding up that process of decomposition. So yeah, summing up 4 years of my life and research, we found that rewilding is a really complex event. It's sort of unrealistic to expect every group to establish. But it's also a really holistic thing, so it can change a lot of different components of the ecosystem depending on what you're looking at. So yeah, it's a cheap tool. It wasn't too hard to do. As I said, it can be done in a day. It's nothing like you have to set up a very big project that takes a lot of time. We did a lot of scoping in terms of trying to find the actual good source remnant community, so that did take a bit of time. But yeah, we're looking at more of the applications of our methods. This idea of soil and litter transplants, they're not really done too much in Australia for whatever reason. In mining projects they are, but it's kind of rare to find this as a restoration method just when we're talking about biodiversity. Overseas is a really different case. So these are some photos from some overseas projects that look at soil transplants in places like California and I think once from Brazil as well. And what these projects are doing is that they're trying to transfer the remnant seed bank from one site into a new site as the restoration method. And as you can see up there, they're sort of doing it at a really big scale as well. So in our project, we were just taking small samples of leaf litter and soil to inoculate a new site. But these projects are essentially replacing the whole soil profile, as well as the seeds and the invertebrates and the microbes. So yeah, these projects are mostly focused on seed transfer, but what they found was that soil transplants are a really cheap and effective way to restore an environment, and a really holistic method too, because you take over the seed and the invertebrates and the microbes as well. So we're really hoping to see, greater uptake of these methods within this country, specifically by councils and restoration practitioners as well. Taking an even broader view, we communicated these results to the public, and we framed it around global invertebrate declines and conservation and rewilding. And we were finding that there was a lot of public appetite for this as a restoration tool, even though it was quite a niche sort of method, when you framed it around this idea of rewilding and trying to reintroduce species into a new area. And that was really promising because we want to see greater uptake of these methods, and it's important to have, the public on your side and understanding what you're trying to do and why you're doing it as well. So So yeah, thank you so much. And I'd like to thank Biolinks for inviting me today and everyone that's helped out with the project so far. So cheers.
Thanks, Peter. We've got time for questions if anyone wants. Yep, okay.
Thanks, great presentation. Really fascinating, very exciting.
Thanks.
Just got a question, I suppose, around did you see of making observations around germination or changes in plant presence in these translocation sites? I've got one site I manage for the council where it's an old capping site, and they've, by serendipity, they brought in impregnated material. So I've got a super rich capping of peas and orchids and all kinds of stuff. Some stuff's blown in, but some of those species, there's no way they blew in. So I was, just curious to see if that is something that was observed, because you're obviously taking leaf litter and invertebrates, but if you're talking about soils, is there you're anticipating that you've got other things should be popping up?
Yeah, we didn't look at germination specifically. So in our methods, we scraped just the top 5 millimetres of topsoil. So we were taking some seeds, but probably not the whole conserved seed bank. But at the same time, we're taking microbes and mycorrhizal fungi as well, which can probably help some germination of some species as well. I've got anecdotal evidence for that, I suppose. Like I took some of the old leaf litter and the soil that was run through the Tolkien funnels and used it as mulch at my house just for my garden, and I was finding lots of different little things popping up as well. So I can imagine that if you take really big volumes of soil, you can see, kind of probably pretty amazing regeneration coming from there, especially in the process of disturbing it as well, which is, I think, important for a lot of native species to pop up too. So, yeah.
G'day, thanks mate, that was fascinating. Did you find any risks of damage, or do you have any guidelines around removal of the soil and mulch from the remnant areas?
Yeah, it's a really good question. So in our case, we didn't take too much from the remnant site, so these were all conservation areas, just small little 80 centimetre by 80 centimetre samples, and that was enough to sort of seed the new site with species. I'm not entirely sure how it would go on on a broader scale. I think some good examples in this country would be, you've got your diverse area that might be slated for development, so they'd come through and they'd clear out all the trees and the bush. I think a lot of that topsoil would just be built upon, or I've heard that sometimes it gets sold to landscaping companies and that sort of thing. So that's sort of like a very big wasted resource that could then be incorporated at other sites. So I think maybe factoring that into some of the development plans at these areas would be a good way to overcome that sort of risk trade. Yeah.
Thanks for that, Ms. Paulson. You mentioned they were mature restorations. Do you have any thoughts on what would be the applicability at time of planting of promoting such conditions?
Yeah, I think at the get-go it would be good as well, because you'd probably bring over some of those microbial species too. We targeted mature restoration because we had such a heavy focus on invertebrates, and a lot of the remnant species are pretty prone to drying out. So if they're transplanted into a very open area, it's probably safe to say that it was too harsh of a condition. So we chose sort of like a more mature site that provided more protection for them. But I don't see any reason as to why you couldn't just take the litter and soil at the start of the planting too, just to sort of seed the community and steer it towards that sort of remnant trajectory as well. But yeah. Questions?
I was just wondering if you've noticed or are aware of any issues of introduced species in the Hackney scale, and if what this idea of translating is into problems that they might be causing, if they are causing problems?
Yeah. So I guess an important thing is that we went from, a more pristine area, and we transplanted that community into a very degraded area. So that degraded area from our baseline samples was filled with invasive species already, so things like there's a lot of invasive springtails and mites because they were ex-pasture. So there's less of a risk when you go that way. It'd be a bit more risky if you went the other way for whatever reason. One thing that always pops up is people's concerns about things like transplanting Phytophthora, but you could just do some thorough DNA metabarcoding beforehand just to understand if that risk is actually present or if it's not there. But yeah.
So sorry, maybe I didn't articulate properly. Is there any benefits in the context of there being introduced species that might be having a negative effect? Are you noticing the benefit of reintroducing predators?
Oh, that was quite hard to determine. We haven't done that yet, but I know a lot of projects, what they'll do is they'll actually strip back the entire community, so they'll scalp the area, so they'll actually get rid of all the introduced species in that area and then dump the soil on top of that. So that's an even better way just to completely remove the in situ population. But yeah.
Thanks again, Peter.